Hello everyone, welcome back. This is week 10 of the course Fundamentals of Protein Chemistry. The course is coordinated by Professor Dasgupta from the Department of Chemistry at IIT Kharagpur, and I am Beas Mukherjee, a research scholar from the Department of Bioscience and Biotechnology, IIT Kharagpur. And in today's class, we are going to talk about protein and its interactions with different macromolecules. For example, we are going to talk about protein-carbohydrate interactions majorly, protein and DNA interactions, protein-RNA interactions. Okay, and we have a special class reserved for protein-protein interactions that we are going to discuss in the next class. So in today's class, let's start with protein-carbohydrate interactions. Okay, so protein-carbohydrate interactions play a vital role in health and disease. Studying these interactions is essential for understanding key biological processes such as cell signaling, immune response, pathogen recognition. These interactions play critical roles in various diseases, making them valuable target for therapeutics, drug design, vaccine development, and personalized medicine. Right. So exploring the field of protein-DNA interactions is vital for understanding gene regulation, DNA replication, and repair mechanisms. So these interactions are crucial for the regulation of gene expression, cellular function, and can influence developmental processes and disease states. Insights from these areas can aid in therapeutic developments for gene editing, personalized medicine, and therapeutics. So we are going to talk about all of these in details. All right. So first, let us talk about omics studies. Now, what exactly are omics? So omics is a very advanced field in which we are studying a lot of different interactions in this at the same time. Okay, it's not specifically focused on a particular gene or a particular protein or a particular RNA molecule, but we are looking at it at a genome level, or let's say a proteome level. So we are going to talk about that. So we know that in central dogma, what happens is the DNA, firstly it undergoes self-replication. So the study of the genome is called genomics. Next comes epigenomics. Now what is that? Epigenomics is actually regulation of gene expression under the influence of different kinds of things. For example, the evaluation of non-DNA sequence changes such as methylation. So there can be different kinds of modifications, post-translational modification on the DNA as well as on the proteins that regulate gene expression. For example, the histone proteins. Okay, so these are the different kinds of modifications. There can be non-histone based modifications, there can be histone based modifications, there can be modifications in the DNA, and all of these different modifications influence gene expression. So genomics deals with identification of genetic variant in DNA sequence, including coding and non-coding regions. Epigenomics deals with evaluation of non-DNA sequences, changes such as methylation. Next is study of RNA. So that refers to transcriptomics. Now DNA gets converted into RNA through transcription, and the study of transcriptomics involves evaluation of quantity and quality of RNA. So this does not include only mRNA but different kinds of RNA, the different kinds of secondary structures that can be formed, the different kinds of modifications, the different kinds of interactions that RNA have. So all of these are included in the study of transcriptomics. Next, when RNA gets converted into protein through the process of translation, the study of these proteins make up the field called proteomics. So evaluation of protein map network, production and modification is included in proteomics. So this deals with the study of proteins at large, the different kinds of modifications that proteins undergo for its proper functioning, the different kinds of interactions that proteins are involved with, let's say with other proteins, with different kinds of small molecules, with different kinds of other molecules. So all of these comes under proteomics. And the next one is metabolomics. So what is that? Now this is at the phenotypic level. So proteins are expressed. Once the proteins are expressed, they are obviously going to perform some kind of function. Okay. And often these functions are related to different kinds of small molecules, metabolites. So they are involved in metabolic pathways, and the study of these metabolites make up the field metabolomics. So this includes evaluation of endogenous and exogenous metabolites. Okay. So we are going to talk about these omic studies in further details now. We look at the definition of omic studies first. I'll go to full screen mode for your better understanding. Okay. So basically, omic studies refers to a large-scale holistic approach to studying biological systems by analyzing entire sets of molecules. Instead of focusing on a single gene or a single protein, omics looks at the collective behavior of thousands of molecules at once. So as we discussed, right? So this is exactly what is said in this definition. Now, when we want to study RNA transcriptomics, now you can understand that if this is not just dealing with a single RNA or a single protein or a single gene, we obviously need a lot of high throughput techniques, right? So what are these some of the techniques that are used for these different kinds of fields? So for RNA transcriptomics, the different techniques that are used are next generation sequencing or NGS. The next one is single cell sequencing, microRNA, microarray. So we are going to talk about all of these different techniques in detail. So these are very important because omic studies are a very interesting it's a very interesting topic, and you can expect a lot of questions on these in your exams. Okay, so please pay attention to today's class. Now, for proteomics. So we are interested in, let's say, studying protein-protein interaction or some kind of post-translational modification of a protein, or the presence and absence of a protein, let's say in some kind of disease condition. So for that, we can apply all of these different techniques. What are these? One is 2D DIGE. This is a kind of gel electrophoresis. Okay. Then there is LCMS, which is liquid chromatography mass spectrometry. Okay. There is Malditov MS. MS is once again mass spectrometry. Then the next one is TMT, and then there is SRM. So you can read about these different techniques. Okay. The next one is DNA, genomics and epigenomics. So if you want to study about genomics and epigenomics, one of the majorly used technique is GWAS. So there is of course DNA footprinting. There is next gen sequencing. Okay, and then there is also single cell sequencing. So these are the different techniques. Then other techniques also come in. For example, CHIP, chromatin immunoprecipitation. These are the different techniques that we are going to discuss in today's class. Okay. Next, for studying radiomics, there are different techniques such as PET scan, CT scan, and MIR, all of these different techniques. Next, for studying metabolomics, we have mass spectrometry, NMR spectroscopy. Okay. And for studying microbiomics, for example, 16S rRNA gene sequencing is one of the techniques used for this. Okay. Right. So now, why are protein carbohydrate interactions even required in the body? So there are different necessities for these interactions. One of this is modifications. So we know that proteins undergo different kinds of post-translational modifications for performing different kinds of functions. So these modifications often involve attachment of carbohydrate moieties. Okay, so what are these different groups that can get attached? These can be acetyl groups, they can be methyl groups, they can be sulfur groups. These can get attached using different kinds of enzymes, for example, esterases and transferases. So one of the important aspects is modifications of proteins. The next one is degradation. So for degradation of different proteins, protein carbohydrate interactions are useful, And the example given to you is glycosyl hydrolases, which is one of the enzymes which are involved in this process. So this can be degradation by chopping off a single amino acid, or it can be on a solid surface. Okay, the next one is interaction and recognition. So this is one of the most important ones. For example, antibody. Now antibodies, these can, let's say, bind to different, these can have different post-translational modifications, and they can be programmed to recognize these and bind to these different carbohydrate modifications on a particular antigen. The next one is lectin. So lectin is a carbohydrate binding protein which is there in the immune system of our body. And then there are chemokines. Chemokines are also important molecules which involve protein carbohydrate interactions. The next one is transport. So we have studied about membrane proteins. We know that membrane proteins are highly selective when it comes to transport of different molecules across the cell, and protein carbohydrate modifications are largely present in the membrane proteins. We have seen that already. So at the peripheral membrane proteins, there are a lot of these protein carbohydrate modifications. Now these are also very important because these are often markers for different diseases. Okay, and that is why it's very important to know them. The next one is synthesis. So glycosyl transferases are important in this process. So protein carbohydrate interactions happen through highly specific non-covalent interactions. So the protein covalent, the protein hydrophilic side is there and the protein hydrophobic side is there, and the carbohydrates are made up of these molecules with OH groups attached, and so these OH groups can perform hydrogen bond interactions with the hydrophilic side of the proteins, and there can also be hydrophobic interactions with the aliphatic amino acids in the protein. So these are the different kinds of interactions. So there can be different kinds of hydrophobic interactions, van der Waals interactions, pi-pi stacking interaction, different kinds of interactions can be involved. Okay, right. So now one very interesting aspect of protein carbohydrate interactions is the blood group. Okay, so we have A antigen and B antigen in the normal ABO blood group system. So there is H antigen which is present in all the blood groups, and then there is A antigen and B antigen which is present in the B blood group and your A blood group respectively. So what are these actually made up of? So H antigen, if you can see that it is made up of these different molecules such as glucose, galactose, N-acetyl glucosamine, fucose, and N-acetyl galactosamine. Okay, so N-acetyl galactosamine is absent in H antigen. In A antigen, N-acetyl galactosamine is added, and in the B antigen, an extra galactose molecule is added. So you can read about this in more details how this exactly works. So now we look at these protein carbohydrate interactions in a little more details. So lectin carbohydrate binding. Okay, and the biological role is actually cell recognition and immune response. So this is majorly associated with viral infections. The next one is glycosylation. So protein stability, signaling, and cell adhesion are majorly the role of glycosylation, and this is involved in cancer and congenital disorders. The next one is selectin carbohydrate interactions. So leukocyte recruitment during inflammation is one of the major aspects of this, and this is largely seen in case of inflammatory diseases, for example rheumatoid arthritis. The next one is sialic acid binding. So this modulates immune evasion, cell signaling, etc. And this is seen in cancer metastasis, viral infections, for example influenza. The next one is heparin and growth factors. So angiogenesis and cell proliferations are the biological role, and this is largely seen in cancer and cardiovascular diseases. Glycosaminoglycans, for example, hyaluronic acid. This is useful in tissue hydration and wound healing, and this is majorly associated with diseases like osteoarthritis, connective tissue disorder, etc. Now, fucose-protein interactions. These are important in cell signaling and adhesion, and they are useful to study the diseases like cancer progression and immune dysfunction. Mannose receptors are useful in endocytosis of pathogens and antigen presentation on the cell surface, and they are associated with infections and immune disorders. So for ABO blood group antigen, blood compatibility and immune response is the function, and they are associated with hemolytic disease and transfusion reaction. So coming to the ABO blood group system, the ABO blood group, actually what happens in case of H antigen is that all the ABO blood groups, they contain this H antigen. Now H antigen is basically modified to make A antigen and B antigen. Okay. Now for A antigen, the only extra thing that we have is glycosaminoglycan. For B antigen, we have H antigen plus an extra galactose molecule that we discussed just now. And for blood group O, it has only H antigen. Okay. Now, blood group A contains the antigen is A antigen and the antibody in the blood is anti-B antibody. For blood group B, the antigen in the blood is actually B antigen and the antibody is anti-A antibody. For blood group A and B, both A and B antigens are present and there is no antibody in the blood. For O antigen, for O group, there is H antigen and there are both anti-A and anti-B antibody in the blood. Okay. Now we'll move on to something known as proteoglycans. So proteoglycans are very interesting aspect. So see, proteoglycans are a class of glycoproteins found in the extracellular matrix. So they are made up of glycosaminoglycan chains that are covalently attached to the protein core. So if you look at this figure, this is the diagram of the extracellular matrix of the cell, and there are collagen fibers, there are proteoglycans, and there are these different molecules that are present. So we can see that the proteoglycans are heavily glycosylated proteins with glycosaminoglycans attached to the protein core. So it is made up of hyaluronic acid and the protein core is there, and the protein core is attached to the hyaluronic acid using a linker protein, and chondroitin sulfate, which is a kind of glycosaminoglycan, and keratin sulfate, these are attached to the protein core, and the globular domain of the protein core is also present. Then we have this entire structure is called the proteoglycan monomer, and the HA binding domain of the protein core is also there. Okay. Now, what is the structure of the proteoglycan? So, proteoglycan is made up of a glycosaminoglycan. A glycosaminoglycan is made up of an uronic acid and an N-acetyl sugar. Now, to this, there are all of these different molecules that are attached to form this tetrasaccharide linker, and the linker is attached to the protein core. Now we come to the first question, which is which among the following is or are true about proteoglycans. So the question is, the first of the options is that they are present in both extracellular and intercellular matrices. Only one sulfated glycosaminoglycan chain is covalently connected to a membrane protein. Glycosaminoglycan is connected to the serine residue through a tetrasaccharide bridge, and heparan sulfate and chondroitin sulfate are the type of proteoglycans. So you have to tell which of these options are correct. Let's look at the right answer. So option number C is correct. One or more sulfated glycosaminoglycan chains can be covalently connected to a membrane protein. Now moving on to more proteoglycans, syndican and glypican. So what are these? So syndicants and glypicans are actually two family of heparin sulfate proteoglycans. Syndicants are transmembrane proteins with a single membrane spanning domain. Glypicans are glycosyl phosphatidyl inositol anchored proteins, which is GPI anchored proteins, and they are attached to the cell membrane via lipid tails. So you can see in the figure on the right is syndicant. The one in the center shows you a glypican, and it is having this GPI linkage, and you can see that there is something known as perlican, which is shown in the right. So how are these made? So they are made up of different molecules of N-acetyl, glucosamine, uronic acid, galactose and xylose. So perlican is made up of only these different molecules. So you can read about them in detail. So there are these heparin sulfate side chains. And when we come to glypicans, glypicans are majorly made up of disulfide bonds. So there are too many disulfide bonds, you can see already. There are disulfide bonds, and there are also perlican-like structure in the glypican. And of course, there is this GPI linkage. Coming to syndicant, we have this conserved domain and variable domain, and there are also keratin sulfate side chain, and there is also these HS side chains, okay, attached to the core protein. Now we have a question on syndicants and glypicans. So syndicants and glypicans are two major families of membrane heparin sulfate proteoglycans. Dash are held by dash interactions between a sequence of dash amino acid residues and the plasma membrane lipids. However, dash are held by covalently attached membrane lipids. So you have to find out what is V, W, X and Y. So we'll look at the answer. So the right answer is option number A, which says that syndicants or glypicans are two major family of membrane heparin sulfate proteoglycans, and syndicants are held by hydrophobic interactions between a sequence of non-polar amino acid residues and plasma membrane lipids. However, glypicans are held together by covalently attached membrane lipids. Okay. So syndicants are held by hydrophobic interactions between a sequence of non-polar amino acid residues and plasma membrane lipids. However, glypicans are held by covalently attached membrane lipids. Now the next question is on glycosphingolipids and the plasma membrane components, in which the dash head groups are dash. So whether it's hydrophobic and a monosaccharide or hydrophilic, monosaccharide or oligosaccharide, that is what you need to find out. So we look at the answer. We see that the right answer is hydrophilic and oligosaccharide. So glycosphingolipids are plasma membrane components in which the hydrophilic head groups are actually oligosaccharide. So how are these made? These glycosphingolipids are actually made up of a sugar unit. To this sugar unit, the sphingosine alcohol unit is attached, and to this a fatty acid unit is attached. Now, there are different kinds of sphingolipids. There can be simple sphingolipids, there can be complex sphingolipids. So, simple sphingolipids include sphingosine alcohol, sphingosine 1-phosphate. There can be ceramides, there can be ceramide 1-phosphate. Coming to the complex sphingolipids. So, in the complex sphingolipids, there is a extra fatty acid group, and there is this phosphocholine group. Okay, so for example, sphingomyelin, which you will find in the neurons. Okay, so sphingomyelin is one of the examples of a complex sphingolipid. Then there can be cerebroside. So cerebroside has a single sugar residue and a fatty acid residue attached to this sphingosine. Okay, and then we have something known as the globoside. So what is a globoside? So globoside has a di, tri or tetrasaccharide residue attached to the fatty acid residue and sphingosine unit. The next one is a sulfatide. So what is that? Sulfatide has a single sugar residue plus sulfate group and fatty acid attached to the sphingosine unit. The next one is ganglioside. So ganglioside is made up of oligosaccharide residue, sialic acid and fatty acid residue attached to the sphingosine unit. Now we move on to lectins. So lectins are a very important group of carbohydrate binding proteins. Why? Because they are found in the complementary systems in our body. Okay, so lectins are proteins that specifically bind to carbohydrates or sugars and participate in cell recognition, cell recognition, cell adhesion, signaling, and immune responses. So they are largely stereospecific and are important in the complement system of the immune system. Okay, immune response. So lectin family, this includes R-type lectin, L-type lectin, P-type, C-type, I-type, and S-type. Now we'll see in details what are these. So the examples are given for you, so you can go through these different examples. Now the major binding specificity for the R-type lectin is galactose or galactose N-acetyl galactose. Okay. The next one is L-type lectin, and the example that is given to you is concavalin A, and the major binding specificity is high mannose. So the structures are also given for your understanding. Okay. Now, if we look at a P-type lectin, so the P-type lectin is majorly made up of this. So it has a specificity for mannose 6-phosphate. Coming to the C-type lectin, it has specificity for mannose, beta-glucan fucose, and glucosamine. Coming to the I-type lectin, it has specificity for sialic acid. Okay, and for S-type lectin, it has a specificity for beta-galactoside. So we'll look at all of these different kinds of lectins in details now. Okay, so what is a P-type lectin? So P-type lectin is actually a phytohematoglutinin or a PHA, and it has binding specificity to N-acetyl galactosamine and galactose, and the functions are cell agglutination and immune response. So examples that are given to you are concavalin A and this PHA. The next one is C-type lectin. So C-type lectin is an example is mannose binding lectin or MBL, and selectin. So mannose binding lectin is important constituent of the complement system of our body, which is an important part of the immune response. And the selectins, E-selectins and P-selectins, and they have the binding specificity for mannose. They are important in mannose, in immune recognition, opsonization of pathogens, and the next one, and it is also important in so selectins. They have specificity for sialylated carbohydrates, and the function is actually cell adhesion in inflammation and immune response. So moving on, we have the I-type lectin, and the example is galactin. So it has a binding specificity for galactose and lactose, and this is involved in cell signaling, apoptosis, and regulation. So another example is calcitonin. So this has a binding specificity for N-acetyl galactosamine, glucosamine. So neuronal signaling and development is its area of activity. So the next one is A-type lectin, and you can read about all of these in details. Okay, so the binding specificity we already know. This is mannose and fucose, and this is involved in immune modulation and cell agglutination. The next one has a affinity for N-acetyl glucosamine and mannose, and the function area of function is antimicrobial activity and immune response. Coming to L-type lectin, lectins from legumes, for example, soybean lectin is one of them, and this has a binding specificity for galactose and mannose, and the functions are cell agglutination, potential therapeutic use, etc. F-type lectins have a binding specificity for fucose binding lectins, and this has a binding specificity for fucose, and the functions are cell adhesion, recognition of fucosylated ligands. Okay, so we have a question for you. So X-type lectins are essential for cell-cell adhesion. Y-type lectins are essential for development and maintenance of the nervous system. Z-type lectins are essential for their role in the generation of functional lysosomes. Identify the types of lectins. So the different options are given. We'll look at the answer now. So the answer is option number D. X is C-type lectin, which is involved in cell-cell adhesion, and Y is essential for the development and maintenance of the nervous system, and this is actually P-type lectin, and Z-type lectin is actually I-type lectin, which is important for the role in generation of functional lysosomes. Now we move on to some of the techniques that are useful for studying these different omics. Okay, so one of them is oligosaccharide microarray. So what happens in oligosaccharide microarray is that the specificity and binding affinity of the glycan-binding proteins, lectins and antibodies are studied for immobilized oligosaccharides. So we can see in the figure that there are these immobilized oligosaccharides, and when we add lectins, antibodies and bacterial and viral proteins, these obviously have affinity for them and these are coming in binding to them. Next, what we do is we perform fluorescence based detection. We can go for mass spectrometry detection and we can go for SPR imaging to find the different kinds of uh, binding affinity and different applications in diseases. So the microarray, high throughput oligosaccharide microarray technology is used in this case, and this microarray based technology has been widely employed for a rapid analysis of the glycan binding properties of lectins and antibodies, the quantitative measurements of glycan binding, glycan protein interactions, detection of cell and pathogens, identification of disease related anti-glycan antibodies for diagnosis, and fast assessment of substrate specificities of the glycoside transferases. Now there's a question on this oligosaccharide microarray. The question is, oligosaccharide microarray are used to determine A. The specificity and affinity of the carbohydrate binding by lectins, amount of carbohydrate bound to the lectin to the lipid membrane, the affinity of a particular amino acid residue to the proteoglycans, and D is the amount of glycation in the protein. So the right answer is A, the specificity and affinity of the carbohydrate binding by lectins. Now there is something known as the HbA1c immunoassay. So what is this all about? So an HbA1c test is also known as a glycated hemoglobin test or a A1C test. This is used to measure the blood sugar levels in the body, which aids in diagnosing and managing diabetes mellitus, okay, which is type 1 diabetes. Now, HbA1c blood test value denoted in percentage. This is an important indicator of the average blood sugar level for the past 2 to 3 months. The HbA1c test normal range includes values between 4 and 5.7%. That is the normal. Okay. Now, less than 6.5% indicates pre-diabetics, and an HbA1c value of 6.5% or more indicates hyperglycemia, which is diabetes mellitus. Now, what is exactly happening in this HbA1c immunoassay? So, we know that hemoglobin is made up of 2 alpha chains and 2 beta chains, and there is this iron and heme, okay, in it. So, what happens is HbA1c is metabolically produced. So, hemoglobin A1c, this is metabolically produced by the reaction of glucose with N-terminal valine residue of each beta chain of the hemoglobin and the subsequent formation of a stable ketoamine. So, this N-terminal hemoglobin beta chain, there is this kind of a cleavage happening between the histidine and leucine residue, and the formation of a stable ketoamine is actually happening. So, pre-treatment and hemoglobin measurement. So whole blood samples are lysed with the hemolyzing solution. Hemoglobin is released from the erythrocytes. The absorbance of the hemoglobin is measured at 570 nanometer after addition of the reagent R1 and is proportional to the total hemoglobin concentration in the sample. Next, HbA1c measurement is done. So after the addition of the reagent 2, which is R2, fructosylated dipeptides from the N-terminal hemoglobin beta chain are released by a protease, and hydrogen peroxide is produced after oxidative cleavage of the fructosylated dipeptides by fructosyl peptide oxidase, which is an enzyme, and H2O2 is determined colorimetrically by the reaction with a chromogen in presence of a peroxidase at 660 nanometer. The absorbance increase is proportional to HbA1c concentration. So hemoglobin A0 is shown to you, and we see that this gives, so hemoglobin A0 is there, and this with glucose gives us Gibbs base. It gets attached to the glucose and gives us Gibbs base, and then hemoglobin A1c is a amidori product. So you can read more about this in details. Okay. Now there is a question on HbA1c test. This is a method to detect a modified form of hemoglobin. Which among the following statements is or are false about HbA1c and or that modified form of hemoglobin? So it is used to measure the average plasma glucose concentration over prolonged periods. It is a form of hemoglobin that is chemically linked to a sugar. It is formed through an enzymatic activity. So which one of these are true? Only 3 is true. HbA1c test is used to measure the amount of blood sugar or glucose attached to hemoglobin, not present in the plasma, but attached to hemoglobin. So this modified form of hemoglobin is actually called glycated hemoglobin, which is formed through a non-enzymatic glycation pathway caused by. Which one of these are actually false? So only 3 is false in this case. The other 2 are correct. Okay. Through non-enzymatic glycation pathway caused by the interaction of glucose with hemoglobin. Now the next question on protein macromolecule interactions is interaction of the protein with the nucleic acid are essential for which of the following biological processes? So DNA replication and repair, RNA processing, translation, gene expression, regulation of chromatin structure. So protein nucleic acid interactions are important for which one of these different processes? This is a very simple question and you should be able to answer this. So DNA replication and repair, of course, uses a lot of different proteins. Important, definitely. RNA processing and translation, yes. Gene expression, yes, we need transcription factors for this. And regulation of chromatin structure. So, of course, there are these different proteins and they undergo different kinds of modifications. There can be histone structure, there can be non-histone proteins. So, histone and non-histone proteins, and they undergo different kinds of chemical modifications for regulation of chromatin structure. So, which one do you think would be the right answer? So, right answer is A, B, C and D. All of them are important for protein nucleic acid interaction mediated applications. So, interactions of proteins with nucleic acid are essential for all the biological processes mentioned above. Okay. Now, we'll look at something known as the helicase enzyme. So, what is there in this helicase enzyme? So, helicase enzyme has an N-terminal region, there is a C-terminal region, and there is the helicase core. Now, this helicase core is made up of Recca-like domain 1 and Recca-like domain 2. So, Recca-like domain 1 has both 1 and 2 have these 3 important functions. One is ATP binding and hydrolysis, another one is RNA binding, and then there is coordination between ATP and RNA binding. So, the dead domain box is characteristic feature of the family of proteins known as the dead box RNA helicase. These proteins are involved in unwinding of RNA, RNA processing and regulation. So, dead comes from the presence of a conserved amino acid sequence within this motif, which is aspartate glutamate alanine and aspartame. So that makes up the term DEAD or dead. Okay. Now, dead box proteins typically contain two A-like domains that facilitate the helicase activity, and dead box helicases utilize ATP hydrolysis to change their conformation and promote unwinding of RNA. Now, another important aspect of protein nucleic acid interaction is the K-homology domain. So, what is that? A K-homology domain is an RNA binding domain involved in the regulation of RNA splicing, translation, and RNA stability. Now, there are two different types of the KH domain. So, KH domain 1 has a KH minimal motif, and then there is a C-terminal region, and then the type 2 KH domain has an N-terminal region, and then the KH minimal domain comes. Okay, so it consists of about 70 to 90 amino acids and adapt a characteristic three-dimensional structure. It consists of two conserved sequence motifs, and it is often referred to as KH1 and KH2, which are critical for RNA binding. This binds to RNA without much sequence specificity. So that is one of the very interesting characteristics of this KH domain. Now we come to the S1 and S2 domains, about which you have studied during the COVID interactions. So COVID-19 virus and its interaction. Okay. So the S1 and S2 domains are RNA binding domains commonly found in various RNA binding proteins, particularly in those involved in RNA metabolism such as splicing, translation, and degradation. So S1 and S2 bind to the single-stranded RNA using their characteristic fold of a beta barrel structure. S2 may also mediate interaction with proteins. So S1 domain and S2 domains are shown in this figure. So they have this S1 domain has this N-terminal domain, then RNA binding domain followed by S1 and S2 domains, and SD1 and SD2 domains, and the sequences are also given for your reference. And this S2 domain has all of these different other domains. Okay, and these are very important in the RNA metabolism such as splicing, translation, and degradation. Now there is a question on this. The nucleic acid binding domain shown in this image is a type of dash. Let's look at the answer, whether this is a dead box domain, an S1 domain, K homology domain, or a S2 domain. So the right answer is a dead box domain. Next we have something called the DNA footprinting assay. This is a very important aspect of studying DNA protein interactions, and it is a very simple strategy but a very useful strategy. So what happens is, there is this labeled DNA, and the DNA is allowed to mix with protein, which is a DNA binding protein, and the DNA binding protein comes and binds to this DNA, and what happens is we perform a digestion using DNase 1 enzyme. So DNase 1 enzyme will go and cleave the DNA, but it cannot cleave the DNA at the points where this protein is bound. So what happens is we get all of these different fragments of DNA and the fragments to which the protein was bound. So we run a gel electrophoresis, and we see that all the regions where the protein was bound, those regions are not appearing on the gel. So that tells us that these are the places where this particular protein goes and binds. So that is how you can know the different proteins that can bind to DNA and the different sequence of the binding site. So which of the following is not true about RNA's footprinting assay? So the same thing, if we do it with RNA, it becomes RNA footprinting. Okay. Now proteins that bind to an RNA sequence can protect that region of the RNA from RNA's digestion. The region that is not protected is known as the footprint of the protein. This makes it possible to locate a protein binding site on a particular RNA molecule. This method uses RNAs to cut the end-labeled RNA with and without bound protein, followed by gel electrophoresis to detect the resulting cleavage pattern. So we look at the right answer. The right answer is not B. Okay. Why? because the region the protein binds to the RNA and the region at which it binds, that is its footprint, not the other region. So definitely option number B is false, and that is the one this question is asking you for. Okay. Now we move on to something known as the replication protein A. Now this is a very important candidate in the DNA repair mechanism, and replication protein A is a protein complex involved in DNA replication and repair. It is a single-stranded DNA binding protein that plays a key role in stabilizing a single-stranded DNA during the various processes such as DNA replication, repair and recombination. So how does it function? It's given in the figure for your understanding. So you can see that this replication protein A, it is useful in case of this homologous recombination and non-homologous end joining when double-stranded DNA breaks are happening, and this protein is useful in once again getting double-stranded form of the DNA. So question on replication protein A, it prevents single-stranded DNA from winding back on itself or from forming a secondary structure. It helps to protect the single-stranded DNA from being attacked by endonucleases. It also binds to the DNA during the nucleotide excision repair process, and it regulates the transcription of a genetic information from DNA to messenger RNA. So which of these is not true about RPA? So D is not true about RPA. Why? Because transcription factors are the proteins which help the transcription of genetic information from DNA to messenger RNA, and this is not a function of RPA. So RPA is basically a single-stranded binding protein which is binding to the single-stranded DNA of the replication fork during its replication. It is also involved in the nucleotide excision repair process, and it is also important in this homologous recombination process for DNA repair. Next we come to CHIP. CHIP is chromatin immunoprecipitation. So what happens is in CHIP is that there is the protein which is bound to DNA, and this is very well cross-linked so that it does not separate from the DNA, and then there is antibody binding, and the antibody binds to the proteins, and then there is immunoprecipitation, washing, and the cross-link reversal. So what happens is the DNA protein analysis happens and DNA purification and quantitative PCR is performed. Now what does CHIP tells us? CHIP tells us the whether a particular protein is bound to DNA or not, and what is the region of binding with that DNA, and this is useful for like omics studies. So cross-linking, so we look at the entire process of CHIP. So cross-linking, now cells are treated with formaldehyde or another cross-linking agent to covalently link proteins to DNA that are bound, and chromatin isolation happens. So the cross-linked chromatin is extracted and then sheared into smaller fragments, typically by sonication or enzymatic digestion. Then immunoprecipitation happens, and antibody specific to the protein of interest is added to the chromatin mixture, allowing the formation of an antibody protein DNA complex. Now this complex is then captured using the protein AG beads, and it is washed. Now the beads are washed to remove the unbound material, enriching for the protein DNA complexes, and then reverse cross-linking happens. So these cross-links are reversed usually by heating or allowing the release of the DNA from the proteins. Finally, DNA purification is done, and this DNA is amplified using PCR and qPCR or sequencing, for example, CHIP sequencing, to identify specific DNA sequences that were bound by the protein of interest. So this tells us that at different points of the cell cycle or cell, different points of the cell cycle, the protein can be bound to different regions in DNA, and this helps in association, studying the association of different proteins with different kinds of diseases, and also the regulation of the overall gene expression. Now there are different applications of CHIP, and there are different methods in which these are used. So one of them is chip on chip. Now here, the first chip is actually chromatin immunoprecipitation, and the next chip comes from microarray. So genome-wide analysis of protein binding sites using microarray analysis of the purified DNA fragments, that is chip on chip. The next one is chip sequencing. So genome-wide analysis of the protein binding sites using deep sequencing of purified DNA fragments. The third one is native chip, which omits the cross-linking step and uses micrococcal nuclease digestion to cut DNA at the histone linkers to examine the DNA target of histone modifying proteins. The next one is chip exo. So addition of an exonuclease digestion step to obtain increased resolution of the protein binding sites up to a single base pair. So these are the different applications of chip. The next one is a pull down assay. So what happens in a pull down assay is, let's say that there is some kind of protein, and this protein is bound to other proteins, or it is bound to an RNA or DNA, and then we have a glutathione affinity resin. So this resin is, let's say, it binds to this particular bait protein. Now there is this bait protein which is bound to it, and the bait protein interacts with the prey proteins. So these prey proteins are the proteins that the bait protein is known to interact with. So this GST tag fused bait protein is immobilized to glutathione affinity resin. After cell lysate addition, the interacting proteins will be bound, and unbound proteins are eluted, and the interacting proteins are pulled down. So all the prey proteins are obtained. So this tells us that, okay, this particular protein has affinity for all of these different other proteins. So that is a pull down assay. Now, question on protein macromolecule interaction. So this question is asking you, which of the following methods is or are used for the detection of protein nucleic acid interaction? So one of them is a pull down assay, electrophoretic mobility shift assay. Now you have already studied about this in one of our previous classes. Then there is tRNA footprinting assay, chromatin immunoprecipitation assay. So we look at the answer. It is 1, 2 and 4. So DNA footprinting assay is used instead of tRNA footprinting assay. So these are the different protein nucleic acid interactions. Now histones have a dash content of dash and dash as well as dash charged amino acids. What is that? Histones have a high content of lysine and arginine as well as positively charged amino acids. Why? Because histones are obviously positively charged, so that they can bind to the negatively charged DNA very tightly. Okay, histones have a high content of lysine and arginine as well as positively charged amino acids. Then we'll talk about the lac operon. Now lac operon is something that we have already discussed in one of our previous classes. You know that there is this promoter region, and very close by to the promoter region is the operating region, and then comes in the structural genes, that is Z, Y and A. They have their different functions and they produce. Z produces beta galactoside enzyme, Y produces permease, and A produces transacetylase enzyme. Right, so now all of these functional genes will be inactive because there is an inducer gene. This inducer, this I gene, lac I gene, this produces a lac repressor protein. This lac repressor protein goes and binds to the lac operator region, and that is why RNA polymerase cannot come and bind to the promoter region. Now what happens is, when the amount of allolactose is very high in the cell, or amount of lactose is high in the cell, that is called the inducer. This inducer molecule comes and binds to the repressor, changes its conformation. It is unable to go and bind to the operator region anymore. So the RNA polymerase comes and binds to the promoter region, and translation of all of these different proteins happen. Okay, now there is a question. So a DNA binding tetrameric protein with 4 identical subunits operates by a helix turn helix motif in its DNA binding domain, which inhibits the expression of gene coding for proteins involved in the metabolism of a disaccharide in bacteria by reducing the transcription of proteins. What is the name of the DNA binding protein? How does the bound repressor reduce the transcription of the specific protein? It is lambda repressor protein. It functions by promoting DNA looping only. It is lac repressor proteins. It functions by only obstructing the RNA polymerase binding site. It is lambda repressor. It functions by prompting DNA looping, obstructing the RNA polymerase binding site. It is lac repressor protein. It functions by prompting DNA looping, obstructing the RNA polymerase binding site. What is happening? Lac repressor is a DNA tetrameric protein that have 4 identical subunits, which operates by a helix turn helix motif in the DNA binding domain. It inhibits the expression of gene coding for proteins involved in the metabolism of lactose in bacteria. So this bound repressor can reduce the transcription of lac proteins by occluding the RNA polymerase binding site or by prompting DNA looping. Now, what is a B-zip motif? Now, this B-zip motif looks like this. It has an N-terminal hydrophilic DNA binding domain for transcriptional activation of the targeted genes. Normally, the DNA recognition sequence is 5' to 3' ACGT, and the C-terminal B-zip has a hydrophobic domain with leucine at every 7th position, stretching on a length of 8 helical turns, tightly held together by 2 alpha helices by around 30 amino acids. This segment is also known as the B-zip or the basic region leucine zipper when it is present in the form of this kind of a dimer. This is a B-zip motif. Now we have a helix turn helix motif. Now what is that? A helix turn helix or zinc. Okay, so now we are going to talk about a zinc finger motif. The next one is a helix turn helix motif. Okay, so what is a zinc finger motif? Once again, I'll make the correction here. Okay, so now this is appropriate. You see, what is a zinc finger motif? Zinc finger motif in a DNA binding protein involves there is a zinc, and the zinc is coordinated by either 4 cysteines or 2 cysteines and 2 histidines. There can also be 1 cysteine and 3 histidine or something like that. Okay, and there is an alpha helix and there is a beta sheet, and this zinc finger motif is actually connecting these two together. So this is very useful in different DNA binding proteins. Now they are asking you a question on different types of DNA binding domains, and you've been asked, so which of these is which kind of motif? So one of them is a helix turn helix motif, and then there are also leucine zipper, and then there is helix loop helix motif, and there are zinc finger motifs. So which one of these are what? So the right answer is B. So 1 is a leucine zipper, 2 is a helix turn helix motif, 3 is a helix loop helix motif, and 4 is a zinc finger motif. So that's all for today. Thank you for your attention. These are the different references that I used for today's class, and happy Pujo to everybody. Celebrate Durga Puja well, or whatever puja it is in your region. All the best. Okay, and we'll meet again for the next class with protein interactions. Thank you so much.